Good morning. My name is Bess Ward, and I'm going to be talking to you about the nitrogen cycle. I know that in your course, you'll be hearing a lot of specific and detailed information about many aspects of the nitrogen cycle and other biogeochemical cycles in the ocean. But today, what I'm going to do is a more general overview and introduction to the nitrogen cycle, and trust that you'll hear a lot of the more interesting details from other people. So I'm going to share my screen and get started. The marine nitrogen cycle is one of the major biogeochemical cycles in the ocean, of course, and it's mainly controlled by microbes. And so that's why I'm calling it the microbial nitrogen cycle, and we'll be stepping through one by one these major pathways that comprise the nitrogen cycle. One of the reasons that the nitrogen cycle deserves a lecture of its own is, of course, that nitrogen is the macronutrient next to carbon that's most abundant in biomass. And it's the macronutrient that's most likely to limit primary production and therefore control all kinds of things that happen in the ocean. Most of you, I'm sure, already familiar with this basic idea that biomass is comprised of carbon, nitrogen, phosphorus, and all kinds of other elements, but the three macro ones in this ratio of 106 to 16 to 1. And that makes it clear that nitrogen is the next most important thing to carbon in terms of the biomass. And so biomass is comprised of, on average, at least, this composition. And in this simple formulation, it's just a carbohydrate and some nitrogen. In a slightly more complicated recipe for biomass, it's carbohydrates, lipids, amino acids, and all kinds of other things, including trace metal cofactors, for example. So nitrogen is an important biogeochemical cycle because of its critical importance in all of biomass and its potential to limit the production of that biomass. It's also really an interesting element because it occurs in a wide range of valent states. And so from the most reduced ammonium at -3 all the way to nitrate at +5. So this is a huge range of electron transfer opportunities for microbes. And microbes are really good at making a living and doing these various transformations and transferring electrons around, and therefore thereby changing the form of inorganic nitrogen that is found in its distribution amongst those different forms. So the two most important ones in terms of nutrition for phytoplankton are nitrate and ammonium at opposite ends of the redox spectrum. The most abundant form of nitrogen in the ocean and everywhere else is nitrogen gas. And that one is important because it's not particularly attractive biologically. And we'll come back to that in a moment. Organic nitrogen, of course, is the one that you're most familiar with because that would be biomass. That would be things like us and everything between us and to bacteria and viruses, for example. So the way I'm going to walk around the nitrogen cycle is using this diagram, which happens to be the one that we have on the back of our lab t-shirt. So it's very useful. We always have it with us. And we're going to step through it piece by piece and put together all the pathways that I just mentioned. And so here are all the pathways that we'll talk about. We're going to go through them one by one and put it back together again. But before I do that, I wanted to mention that important distinction between nitrogen gas and all the other components. If you look at this cycle, it's mostly inorganic nitrogen or dissolved inorganic nitrogen, DIN. All of these forms are inorganic, and here's that organic nitrogen pool right here that represents biomass. Another way to split it up, though, is to think about everything except nitrogen gas. And all of these other forms of fixed nitrogen are what we consider fixed, so they're biologically available. And that is distinct from N2 and N2O, which are the two things that are not fixed, and they're distinguished from all the other inorganic forms of nitrogen by having nitrogen-nitrogen bonds, which are very stable, difficult to break, and so only a few different kinds of bacteria can actually do that. And so that is a really important distinction, biologically fixed or the opposite, not fixed nitrogen. And it's such an important distinction that we actually think about nitrogen budgets in terms of fixed versus non-fixed nitrogen. And so if you make a nitrogen budget, you're usually talking about the total sources and sinks for fixed nitrogen. So look at the information in the two red boxes. These are now 3 different budgets by 3 different people over time. And they compute the sources of fixed nitrogen, which all come from nitrogen fixation, which I'll talk about in a moment. And nitrogen fixation then creates fixed nitrogen, the stuff that all the other organisms can use. And the total sinks, which we'll talk about in terms of denitrification and Anammox. And as you see from these 3 budgets, that the first 2, this one from the late 90s, I think, this one from 2004, and this one from 2008, the first 2 show that the loss terms exceed the input terms. And this seems like an important idea, if true, because that would mean that the ocean would run out of fixed nitrogen, and the organisms that depend upon nitrogen for that might limit the productivity, in other words, would run out of nitrogen eventually. If you think about the sort of total fixed nitrogen inventory of the ocean, the deficit of this scale would mean something like a few thousand years, and then you'd run out of fixed nitrogen. So there's a kind of a suspicion that it's not true, that it really isn't out of balance that much. And Gruber's more recent budget says, well, it's actually pretty much in balance. But a lot of measurements implied that denitrification outweighed nitrogen fixation. And so this inspired a lot of work on the nitrogen cycle to quantify both of those processes in an improved way. And you can imagine it's hard to come up with a global nitrogen budget because you cannot measure these processes everywhere all the time. So there's a lot of uncertainty that may or may not be represented in these budgets. But the potential for the budget to be out of balance, that you might be losing more fixed nitrogen than you were producing, led people to be very interested in finding more sources of nitrogen fixation. And so that's been a big driver in nitrogen cycle research in the last few years. So we'll start with the first process in that nitrogen cycle, nitrogen fixation. So to wrap up nitrogen fixation, it's energetically very expensive for the cell to do. It's highly regulated for that reason. It's regulated by light. If they have dissolved inorganic nitrogen present, sometimes they will not fix nitrogen, although that's not a hard and fast rule. And the nitrogenase enzyme is very sensitive to oxygen, so that poses a dilemma for cyanobacteria who make oxygen doing photosynthesis. The ability to fix nitrogen is restricted to bacteria, especially in the ocean to cyanobacteria are the main ones. You find the highest rates in the tropics and subtropics, and importantly, it's the only biological source of fixed nitrogen. And that's why it's important in terms of the nitrogen budget that we just spoke about. So now we have the nitrogen fixed into a form that organisms can use. What happens to it next? Well, it gets assimilated by phytoplankton and bacteria. That's the main organisms that we're interested in, how they get their nitrogen at the base of the food chain. And so we're looking at this arrow now where we have organic material and ammonium being assimilated into the organic matter and also being remineralized. So this is just the forward and reverse reaction. So we'll come back to that. So ammonium is one of the main sources of nitrogen for phytoplankton, but nitrate is the most abundant form of nitrogen in the ocean, and phytoplankton definitely use that too. So assimilation of nitrogen by phytoplankton is usually you focus on nitrate and ammonium both. So ammonium assimilation, nitrate, nitrite assimilation, and also amino acids. So I'm mainly concerned now with phytoplankton, some of whom take up amino acids. Assimilating amino acids is definitely a good thing to do because it's ready to slot right into a protein, but most phytoplankton don't get most of their nitrogen that way. So the best way and the easiest way to assimilate nitrogen is to use ammonium, and just about every kind of phytoplankton can do that. I don't know of any that can't. So this is just a slide that is an opportunity to show you some beautiful phytoplankton. These are mainly diatoms, but there's also Serratium here, which is a dinoflagellate. And a huge variety of phytoplankton and bacteria assimilate mainly ammonium as the favorite, although not always the most abundant nitrogen source. So the simple way to assimilate nitrogen is to get ammonium. And that's very straightforward to, in a one-step reaction, to incorporate it into alpha ketoglutarate, which is a 5-carbon backbone dicarboxylic acid. And if you add 1 ammonium to it, you get the amino acid glutamate. And the enzyme that catalyzes that reaction is called glutamate dehydrogenase, or GDH. And that was the first way to assimilate ammonium that was figured out. The people figured out that bacteria could do it. But it turned out that that's not the way that most organisms do it in the ocean, because it's not a very high affinity system. You have to have a lot of ammonium. And ammonium is not very abundant in the ocean, mainly because everyone likes it so much. But to understand how we actually do assimilate, how phytoplankton do assimilate nitrogen in the ocean, we still have to understand the chemistry of this reaction. So what we saw previously was that alpha ketoglutarate was the molecule onto which we add this ammonium in order to get it incorporated into organic nitrogen. Here's alpha ketoglutarate. It's that 5-carbon dicarboxylic acid, has no nitrogen. You add 1 amino group to it, you get glutamate. So that's the same backbone, now with 1 amino group. And if you add 2 amino groups to it, you get glutamine. And so the transfer of amino groups between these 3 compounds is the way that phytoplankton assimilates nitrogen. And this turns out to be a much more complicated but much higher affinity pathway that's similar to what we just saw, but now is incorporated with a much more complex and high, as I said, high affinity. In other words, works well at low ammonium concentrations. So the first step is the glutamine synthase enzyme that adds this ammonium group onto a glutamate molecule. So that was the carbon backbone with 1 amino group. We add another amino group to it, we get L-glutamine, which has 2 ammonium groups. And then you transfer 1 of them to alpha ketoglutarate, which has none. So you go from 1 molecule that has 2, gives 1 of them to alpha ketoglutarate, you come up with 2 molecules of glutamate. And then that glutamate can then hand that nitrogen group off to some other amino acid or some other metabolic purpose and regenerate the alpha ketoglutarate. And so you start with ammonium, inorganic ammonium, transfer it to glutamine and then to glutamate, and then on to the rest of the nitrogen cycle in the cell. And this is also the basis of the way that phytoplankton assimilate other forms of nitrogen. So if you don't have ammonium, but you have a lot of nitrate or nitrite, then those can be assimilated through the same pathway, basically. First, you simply reduce the nitrate to nitrite and reduce the nitrite to ammonium, and then you can take it through the regular GS-GOGAT pathway that we just talked about. And the reason that ammonium is preferred is you can see that one, ammonium is at the same redox level as organic material, whereas in order to assimilate nitrate and nitrite, you have to reduce them first. And reducing power is energy, is an energy requirement. And so it's more expensive to assimilate nitrate and nitrate, but you assimilate them by basically putting them into the same pathway as ammonium. You just have to reduce them first. And so the distribution, I'm not going to have time to show you much about the distribution of all of these processes, but just to say a few words, the most obvious thing is that nitrogen uptake, nitrate uptake, and ammonium uptake by phytoplankton mainly occurs in the surface layer. And that's, of course, because that's where the phytoplankton are. But the surface layer is pretty thin. And so these are a bunch of stations, a bunch of visits to similar stations in the Northeast Atlantic. And they just show that whether it's winter or summer, you get a pretty clear distribution of nitrate assimilation being most rapid in the surface and declining to depth, so that there's no sink for nitrate, at least not very much, below about 100 meters, right? Not below the photic zone. So there we have nitrogen assimilation, and it's mainly occurring in the very surface layer of the ocean. Bacteria, of course, assimilate nitrogen as well, but it's not a net sort of reaction because they aren't making, it's not a net sink because they aren't doing photosynthesis and creating new biomass. They're recycling and mostly heterotrophs. So the other part of that reaction, the opposite reaction, was remineralization. So if you assimilate ammonium into biomass, the backwards reaction of that is just ammonium remineralization or ammonification. And all that is turning organic matter back into ammonium. And that's called remineralization because we're taking it back into the original mineral form. And that's just an example of remineralization in general, which heterotrophic bacteria and zooplankton, for example, do that process. We're back to our general recipe for biomass here. And what happens is that you start with the biomass of that average recipe. And if you decompose it in order to consume food, to respire oxygen, that results in remineralization of these compounds back into their inorganic mineral forms in the ratio in which they were present in biomass. And so that's a really important concept in oceanography, that you basically have average biomass of the same sort of average composition. And if you decompose it, you get back what you started with in terms of inorganic nutrients. So that's a really important concept in oceanography, that if you start with an average kind of biomass and consume it for food and energy, respiring oxygen in this case, then you remineralize and regenerate those elements back into their original mineral form in the same ratio that they occurred in the biomass. So in the case of nitrogen, we've remineralized the biomass and we now end up with ammonium. So what's the next step in the nitrogen cycle after that? That's nitrification. And that is the process of oxidizing ammonium first to nitrite and then to nitrate. And this is performed by nitrifying bacteria and archaea. And all of these organisms are autotrophs, so they're fixing CO2. It's a very hard way to make a living. Not a whole lot of energy is gained from oxidizing ammonium and nitrite, but enough to support the biomass of a lot of bacteria and archaea in the process of oxidizing ammonium to nitrite and nitrite to nitrate. So the first step is best known in bacteria, and it's another of those cases in another one of those situations in which we know a lot about how it's done in some organisms, but they turn out not to be the ones who are doing it in the ocean. So ammonia oxidizing bacteria, AOB, take it through various steps, ending up with the consumption of 1.5 moles of oxygen in order to oxidize ammonium to nitrite. Here's nitrite. And the most important AOB in the ocean are Nitrosus spira. You can see why they're called spira. The actual clades or strains that we think are important in the ocean are not actually in culture, but these are some of their close relatives. And so these have been known and studied in soils and in the ocean for over 100 years. In the ocean, in the early 70s, a lot of AOB were isolated and studied by Stan Watson. But it turned out in the early 2000s, it was discovered that ammonia oxidizing archaea are actually the organisms that do most of the ammonium oxidation in the ocean. And these are, again, tiny little guys. These are a micron or less. And they were discovered in a fish farm, an aquarium in Seattle. And they basically use most of the same machinery, but we don't really know the pathway by which they oxidize ammonium. They have the same basic enzymes, but it's a completely different metabolism. And so we don't really know the mechanism by which ammonium is oxidized to nitrite by these guys. But we know that they do it. They do it stoichiometrically. Here's the growth in the number of cells over time over 3 weeks, right? Hard way to make a living. It takes 3 weeks to get that far. The ammonium that's added is stoichiometrically converted to nitrite that accumulates in the medium. So there was some doubt that these guys were doing it, but they are the ones who are most important in the ocean. And you can see in the abundance of the crens, which are the AOA and the beta AOB, that there are thousands more AOA than there are AOB. So it's clear that they're the ones who are really abundant in the ocean and doing all the work. And they typically show this sort of subsurface maximum distribution, and the ammonium oxidation rate usually shows the same kind of distribution. We'll come back to that in a moment. So ammonium oxidation results in the production of nitrite. There are organisms that have very recently been discovered who can oxidize ammonium all the way to nitrate, but they haven't been found in the ocean so far, which is really, really interesting. I'm just waiting, but so far they have not. So if you get to nitrite, then the next step is nitrification. And the second step of nitrification is nitrite oxidation by nitrite oxidizing bacteria. And we think that's a little simpler to go from nitrite to nitrate. It's only 2 electrons, not like 6 for ammonium. We think that the most important nitrite oxidizers in the ocean are relatives of nitrospina. So this is another isolate from Stan Watson. That's not the exact clade, but it looks like some of their close relatives are the most important nitrite oxidizers, most abundant nitrite oxidizers in the ocean. So I alluded to the distribution of nitrification in the ocean, and it generally shows the opposite kind of distribution compared to nitrate assimilation. So we saw this nitrate assimilation profiles in the same stations in the same study. Nitrification was measured, and this is measuring the production of nitrite and nitrate. And you see that it's sometimes you find it in surface waters, but mostly it's pretty minimum right near the surface, and it increases with depth. And that's a very classic distribution for nitrification, that it's minimal in the surface, and there's a subsurface maximum right about the place where phytoplankton stop taking up nitrate. And so the distribution of nitrification throughout the entire ocean looks something like this. If you already have we didn't measure it in the surface because we knew it was fairly minimal there, but you have a strong peak here, and then it decays very rapidly with depth. And this is interesting because for a long time, people knew that the ocean was full of nitrate. So if this is the distribution of nitrate, it's minimal in the surface and then increases with depth. This is a global ocean average nitrate distribution. And so it was assumed that nitrification happened down here. But in fact, the maximum nitrification rate is very near the bottom of the photic zone. And so the reason that the ocean is full of nitrate is that there's no sink term there, right? The phytoplankton assimilating nitrate mainly do it in the surface. And so everybody's fighting over nitrate at the bottom of the photic zone. Below the photic zone, there's no sink. And so it accumulates. And so what you see is that this actual total depth distribution of nitrification, which leads to the production and accumulation of nitrate, is basically the last step in mineralization, returning the nitrogen to the original mineral form, has this distribution that looks very much like the Martin curve, with which I suspect you're already familiar. And this just shows one of the first compilations led to them being named the Martin curve, in a paper by Martin et al. And these are sediment trap fluxes that were measured at different depths in the water column at a bunch of different stations. And you see that the flux decays with depth. So most of the flux you catch right under the surface layer where the phytoplankton are. And then if you go down to 500 meters, you only catch this much. And that means that this difference in what you catch in the surface and at 500 meters is the material that has been remineralized in that depth interval. And so that makes total sense when you think about the distribution of nitrification, because that material that is decomposed, remineralized, shows up as ammonium, it's nitrified. So everything in the ocean, or at least most things in the ocean, show a distribution that can be related to the Martin curve, and the idea that everything happens in the surface ocean, right? All the fast turnover production, net production, and fast turnover happens in the surface ocean, and then everything decays with depth. So nitrification is accomplished by autotrophic bacteria and archaea. They are generally light inhibited. I didn't mention that that is true, but they are physiologically light inhibited. It damages their cells, although the mechanism is not entirely clear. But it's true of both bacteria and archaea. And they are the ones, as I said, that complete the process of mineralization of nitrogen in the ocean, leading to the fact that the ocean is full of nitrate because there is nobody down there to assimilate it. So now we've got all the nitrogen turned back into nitrate. What do we do with it next? That's denitrification. Denitrification literally takes the nitrate all the way back to N2 gas. And it's a multi-step process that we thought for a long time was accomplished by regular conventional denitrifying bacteria that did the whole thing. And the reason that denitrifiers use nitrate, nitrite, NO, and N2O is they use these compounds as alternative electron acceptors, alternative respiratory substrates. So instead of respiring oxygen to break down organic matter, they use nitrate. So they still break down organic matter, and they're therefore heterotrophic bacteria for the most part. And they consume the organic matter and release some of that remineralized nitrogen themselves back into ammonium. And so this is called, referred to as canonical or complete denitrification, assuming that we start with nitrate and sequentially use each of these substrates. We reduce nitrate to nitrite, nitrite to nitric oxide to N2O to N2, using each of them along the way as an alternative electron acceptor, with the net effect being that you produce N2. And remember, that means that it takes it back out of the fixed nitrogen inventory. It turns out, though, we learned this partly through the molecular studies that have been done on denitrification, that denitrification is actually usually modular. In other words, it turns out to be fairly rare that the organisms that perform denitrification in the ocean do the whole process. Most of them actually do this step, or they'll do this set of steps, or they'll do this step. But only the lab rats in culture basically do the whole thing. Very rare in terms of the abundance and distribution of denitrifiers. It turns out most of them are modular. And the reason that they do both of these here is that NO is a toxic, dangerous chemical. And so if you're going to make NO, you're going to make N2O. You're going to get rid of NO one way or the other. And it's not just bacteria anymore that do denitrification. There are forams, and then most of there are, as I mentioned, this modular idea, there are many different kinds of denitrifying bacteria that do different pieces of the process, have all kinds of different other kinds of metabolism associated with their cells. It's a huge diversity of organisms. But there are also denitrifying forams. These mostly live in sediments, so they perform denitrification in the sediments, and so far, they've only been shown to take nitrite to N2O, but that is removing the nitrogen from the fixed nitrogen budget. So they're important denitrifiers in sediments. And that's a very currently developing area of study, how important eukaryotic denitrification is. So denitrification is the main pathway by which nitrogen is lost, that is converted back to unfixed nitrogen, to N2 in the ocean. But there's another pathway that turns out to be very important, and again was only discovered recently, relatively recently in the 90s. And that is Anammox. So Anammox takes ammonium plus nitrite and turns it into N2. And so they have the same effect on the nitrogen budget, sort of the same role in the nitrogen cycle by turning DIN back into N2 gas, which is not available, not biologically available. So that's a really important function that they have, but they're completely different organisms than denitrifiers. There's only one group, one clade that's known to do it, Anammox bacteria and the planktomycetes. And they're autotrophs. They fix their own CO2. So in that sense, they're kind of like nitrifiers. They call it anaerobic ammonium oxidation. And so they produce both nitrite and N2 gas. And it was really exciting when these guys were discovered, because they're really strange organisms. They have never been isolated in pure culture. They seem only to grow well with their friends. They have a lot of close friends. The genome that was sequenced was one of about 28 or 28, maybe 39, something like that. Different kinds of organisms were present in the culture, as pure as they could get it. But they were the main part of the biomass, and so they were able to sequence the whole genome. So they're shown here as they are bright red because they are so full of heme, so full of cytochromes, and a lot of iron. And so this is granules in a bioreactor. So that's a big size, right? 1 centimeter. So there's lots and lots of cells here. This is one individual cell in TEM, and you can see one of the things that's weird about them is that they have this internal membrane structure, which is very unusual in bacteria. And it's called the anammoxosome. So here's a diagram of it. And it's thought that the Anammox reaction, the anaerobic ammonium oxidation reaction actually happens inside this internal membrane-bound structure, which has unique and interesting membranes associated with it. And one of the intermediates is hydrazine, which is rocket fuel. And so it's sort of assumed that this is kind of a dangerous intermediate, like NO, only worse. And so it's sequestered inside the cell in the anammoxisome. And so when Anammox was discovered and first measured, it was realized that we didn't know how important it was compared to denitrification. Had we been measuring into production and blithely thinking that that was denitrification? So it wasn't really until Anammox was discovered that people came up with a good way to actually measure denitrification. And so this again depends upon an N15 tracer. Usually we use nitrite, and you add N15 as the tracer and measure its accumulation in N230 gas. To distinguish that from ammonium, though, you add N15 ammonium label and then measure that accumulation in N29. And so those are the two reactions that you use to distinguish and to measure them both independently. And the breakthrough that the Danes made in figuring out this method was, the reason it's hard is that seawater is full of nitrogen gas, it's saturated with nitrogen gas. So if you add your tracer as nitrate, it's really hard to pick it up and to get the sensitivity to measure it in the nitrogen pool, in the dinitrogen pool. And so what the scientists from Denmark did, Dalsgaard and Vaundrup, was that they figured out that all you had to do was strip the nitrogen gas out of the tubes first and then add the tracer, and then you could measure the accumulation of nitrogen gas. So this is just our setup on one of our cruises. It's pretty straightforward. You use these tiny little vials, flush them, add your tracer, measure its accumulation into gas. And so when you do that, you find that the distribution, that both ammonium and, both Anammox and denitrification occur often in the same places, not always in the same vials. They are clearly regulated by different things, the heterotrophs versus the autotrophs, the requirement for nitrate versus ammonium. But what we found was that you could predict the relative rates of the two processes on the basis of what organic matter was being decomposed. Remember, the denitrifiers are heterotrophs. They require the presence of organic matter. If you didn't give them any organic matter, then you got this kind of rate of denitrification in the white bar, Anammox in the black bar. If you gave them sugar, that's something that has no nitrogen, then Anammox was not increased, but denitrification was. So they could live, at least for a while, by just consuming sugar. If you gave them amino acids, both were increased. If you gave them particulate organic matter that you had collected that day in the ocean, in the same water in which these guys had been growing, then they were both stimulated. And if you calculate the ratio that you expect between Anammox and denitrification on the basis of the C to N ratio of the substrate, that's the gray bar, and you can see that that predicted the measured Anammox very well. And the reason for that, of course, is that Anammox requires ammonium and nitrite. And there is basically no ammonium and nitrite in the ocean for the most part. It's really, really low concentrations. And so in most of the ocean, the place that you would get ammonium and nitrite would be through the decomposition of organic matter, the remineralization process that we spoke about. And if you're in an oxygen minimum zone, I forgot to mention that both denitrification and Anammox are obligately anaerobic processes. So the only place that they occur is when there is no oxygen. And when there is no oxygen, then you don't have aerobic respiration doing the remineralization. And so the only process that can be active in oxygen minimum zones when there is no oxygen in order to produce the mineralization of ammonium, to produce ammonium and nitrite, is denitrification. So you've got denitrification doing double duty here. They're decomposing the organic matter, respiring nitrate, producing N2, and also producing ammonium and nitrite that can then be consumed by Anammox. And so you can't have Anammox without denitrification, at least somewhere in the ocean. They don't have to be doing it all together at the same time, but they have to be coupled. And here's the reaction for Anammox, which shows that you use ammonium plus nitrite, fix a little tiny bit of CO2 to produce some nitrogen gas and some nitrate, because peculiarly enough, the energy source for this autotrophic reaction is the oxidation of nitrite. So we can see why tracer experiments are necessary to tease this all apart. So the distribution of these things on denitrification and Anammox is restricted to oxygen minimum zones, but they also show the same kind of pattern that we just saw for nitrification, in that it's usually distributed as something that looks very much like the Martin curve, because again, they are doing the remineralization, right? They're linked to the remineralization of organic matter. And so these are the sums of denitrification and Anammox with depth in this location off Peru. And it scales very well to something that looks like a Martin curve. And so that was sort of a mystery solved, that the Anammox has to depend on denitrification. And so if that were the only thing involved, you would always get about 71% of the N2 loss, the nitrogen loss into N2 would be due to denitrification, and 29% would be Anammox. And sometimes that's exactly what you find, as in the slides that I just showed you. Other times you find that there's more Anammox than there should be, and that probably relates to either an allochthonous source of ammonium or some recycling of modular denitrification in the near surface layer. And that's something that is actively under investigation now. So that's all I really want to say about the nitrogen cycle in terms of we've made our way through the major processes that are involved. But I did want to bring up one other interesting facet of it, which we sort of skipped over. We mentioned that nitrous oxide was one of the steps in the denitrification pathway. So here's the denitrification pathway: nitrate, nitrite, NO, and N2O. And nitrous oxide, not only a form of unfixed nitrogen, but it's an incredibly strong radiative forcing greenhouse gas, and it's an ozone destroyer. And so it's kind of an interesting gas to consider its sources in the ocean. And so I'll just show you a little bit about that as a facet of nitrification and denitrification. So this is what the distribution of nitrous oxide looks like in the ocean. There's usually a subsurface peak, and then there's a minimum. This is when you are in an oxygen minimum region. So there's an undersaturated region of nitrous oxide, and then it increases, and then, like everything else, decreases with depth. So if we were plotting this in a non-OMZ region, we would have pretty much a straight line like this. So nitrous oxide is really dynamic in oxygen minimum zones. And so the question is, does any of this nitrous oxide make it out to the surface, across the surface and into the atmosphere? Is the ocean a source or a sink for nitrous oxide? And we think it's probably a small source. Here's some results of experiments in which we actually measured the consumption of nitrous oxide and calculated the production time, the production term. I think I'm going to go straight to the little model that shows that nitrification is a production term for nitrous oxide right near the, like below the photic zone at the top of the oxygen minimum zone. Denitrification is a net source right there at the top of the oxygen minimum zone. And then throughout the deep water, denitrification is a net sink. And so what this means is that there is net nitrous oxide production in the oxycline, which can be within 100 or so meters of the surface, and also can be shoaled up to the surface area via upwelling, for example. And so nitrous oxide is a trace component of the nitrogen inventory, and a transient step in the denitrification pathway does comprise a net source of nitrous oxide to the atmosphere. So let's just wrap up by looking at the distribution of the nitrogen cycle in the ocean. And this is a diagram from Kevin Arrigo showing just the vertical distribution of all those processes that we just talked about. So here's nitrogen fixation, primarily in the surface because it's primarily cyanobacteria. Nitrate assimilation is primarily phytoplankton. Nitrite assimilation as well. Ammonium assimilation is bacteria and phytoplankton. Some bacteria assimilate nitrate, but that's predominantly phytoplankton. Here's the nitrification reaction, taking ammonium back to the more oxidized forms of nitrate. Then when you're in an anoxic situation, like in an oxygen minimum zone, we can have both denitrification and Anammox. I didn't mention DNRA because it's not generally very important in the water. It's important in sediments, but not very important in the water column. And here's our sinking particulate organic matter, remember the Martin curve, and it's a modification and remineralization. So just to give you a spatial orientation of the nitrogen cycle, here's another one of those that just, again, shows you where things happen and emphasizes, again, that everything happens in the surface. The net input of organic matter and therefore fixed nitrogen is in the surface, and then various forms of mineralization and alternative respiration determines the distribution of DIN in the deep water. So there you have it. I'll take you back to the nitrogen cycle as the summary of the processes, and I look forward to talking with you individually or separately and answering questions. Thank you.